All right, so today we're going to be diving into the world of T-cell checkpoints. And I think, you hear a lot about this kind of thing in cancer therapy and in immunology. So it'd be really cool to get like a deeper understanding.
Yeah,
And
absolutely.
like how it all works.
It's a fascinating area, and like you said, a really important one for cancer immunotherapy. So yeah, happy to dive in.
Cool. So T-cells, right? I mean, these are like the soldiers of our immune system. And they're constantly patrolling, looking for things that shouldn't be there. And, that's how our bodies fight off, infections and even cancer. But how do they actually, get activated? Like, how do they know when to attack?
Yeah, that's a great question. So they need a couple of signals to actually become fully activated. The first signal is through their T cell receptor. So this is a protein that's on the surface of the T cell, and it's able to recognize a specific antigen. Now, an antigen is just basically anything that the immune system can recognize as foreign. So that could be a piece of a virus or bacteria or even a cancer cell. And the T cell receptor binds to this antigen. That's the first signal. And it's kind of like the T cell saying, okay, I see something that's not supposed to be here.
Okay.
But that's not enough to actually launch a full-blown immune response. They need a second signal. And the second signal comes from what we call co-stimulatory receptors.
Okay.
So these are other proteins that are also on the surface of the T cell. And they bind to ligands on other immune cells. And one of the most important co-stimulatory receptors is called CD28.
So CD28, it sounds familiar.
Yeah, it's a big player in the world of T cell activation. So when CD28 on the T cell binds to its ligand, that provides the second signal. It's like the T cell is getting confirmation that, yes, this is a real threat. We need to mount a full-blown immune response.
So without that second signal from CD28, the T cell is basically like, wow, I'm not really sure if this is a big deal or not, so I'm not going to, go crazy.
Exactly. It's a way of preventing the immune system from overreacting and potentially causing damage to healthy tissues. You need that double check, that first and second signal to really get things going.
Wow, that's fascinating. So, we've got the go signal, but what about the stop signal? How do we prevent these, super soldiers from going overboard?
Yeah, that's where things get even more interesting. So we have these other receptors called co-inhibitory receptors, and these are basically the brakes on the immune system.
Oh, cool.
They help to keep the T cells in check and prevent them from attacking healthy tissues. And two of the most important co-inhibitory receptors are CTLA-4 and PD-1.
Right, those are the ones you hear about a lot.
Yeah.
It's the checkpoint inhibitors.
Yeah, exactly. So CTLA-4 is actually quite similar to CD28. It binds to the same ligands, but instead of activating the T cell, it actually inhibits it.
Oh, wow. So they bind to the same thing, but they have opposite effects.
Yeah, it's kind of counterintuitive, but it's a really elegant way of controlling the immune response. So you have these two opposing forces, the co-stimulatory signal from CD28 and the co-inhibitory signal from CTLA-4. And the balance between these two signals determines whether the T cell will become activated or not.
That's so cool. And what about PD-1?
PD-1 is a little bit different. It binds to a different ligand, and it's thought to play a role in what we call immune exhaustion.
Okay.
So when T cells are exposed to an antigen for a long period of time, they can start to express PD-1. And this is a sign that the T cells are getting tired.
They're getting burnt out.
Yeah, basically.
Right.
And when PD-1 binds to its ligand, it sends a signal that tells the T cell to shut down.
So it's like a timeout.
Yeah, you can think of it that way. It's a way of preventing the T cells from becoming overactivated and causing too much inflammation.
So we've got CD28, which is like the gas pedal.
Right.
And then we've got CTLA-4 and PD-1, which are like the brakes.
Exactly. And it's this interplay between the gas pedal and the brakes that really fine-tunes the immune response.
And you know what's so fascinating about this is that cancer cells have actually figured out how to exploit this system. They can express ligands for PD-1.
Yeah.
And basically tell the T cells to leave them alone.
Yeah. It's really remarkable. They've evolved ways to essentially put the brakes on the immune system.
And evade destruction.
Wow. So they're like, hey, everything's cool here. Nothing to see here.
Exactly. And this is one of the reasons why cancer is so difficult to treat.
That's incredible. So, this whole system of checks and balances is really amazing. But I understand that there's even more to this story, right? There's this whole other layer of control that happens within the cell itself. Yeah, you're right. So it's not just about the receptors on the surface of the cell. It's also about how these signals are processed inside the cell.
Okay.
And this brings us to something called microdomains.
Microdomains.
Yeah, these are tiny compartments within the cell. And they act like little signaling hubs.
So it's not just like a a big soup where all these molecules are just floating around randomly.
Right. There's actually a lot of organization within the cell.
Fascinating.
And these microdomains help to ensure that the right signals are being sent to the right places.
Okay.
And one of the key players in organizing these microdomains are these proteins called A-kinase anchoring proteins.
Or AK pigs.
Yeah, you got it.
Okay.
So AKPIBs are able to bind to a variety of different signaling molecules, and they help to bring them together in these microdomains.
They're like the party planners.
Yeah, that's a good analogy.
They're making sure that the right guests are at the right tables.
Right. And this is really important for ensuring that the signals are being transmitted efficiently.
Wow. So we've got these, checks and balances with the co-stimulatory and co-inhibitory receptors. And then we've got these microdomains organized by AKPs. It's like a whole symphony of signals happening inside the cell.
Yeah, it's really remarkable. And it just goes to show how complex and sophisticated our immune system really is.
This is mind-blowing.
Yeah.
And I'm really eager to learn more about how these checkpoint inhibitors work and what the future holds for immunotherapy. But for now, let's take a step back and delve into some specific research that highlights the intricate world of thyroid hormone receptors.
Sounds good. They're another fascinating example of how cells use signals to control their activity.
All right, so let's dive in.
Yeah, so thyroid hormone receptors are really fascinating. They play a really important role in regulating a lot of different processes in the body, everything from metabolism to growth to development. And they do this by binding to DNA and controlling which genes are turned on and off.
Oh, wow. So they're like little light switches.
Yeah, kind of. They bind to specific regions of the DNA called thyroid hormone response elements. And when they bind to these elements, they can either activate or repress the genes that are nearby.
So it's like they're reading the sheet music.
Yeah, exactly. And the thyroid hormone itself, it's like the conductor. It's what tells the receptors what to do.
Okay, so the thyroid hormone is produced by the thyroid gland, but the receptors are in the nucleus of the cell. So how does the hormone actually get to the receptors?
That's a good question. So thyroid hormone is actually a very small molecule, and it's able to pass through cell membranes. So it can actually travel directly from the bloodstream into the nucleus of the cell.
Oh, wow. So it's like it has a backstage pass?
Yeah.
It can just bypass all the security?
And once it gets into the nucleus, it binds to its receptor. And that causes a change in the shape of the receptor. And that change in shape, it's what allows the receptor to bind to DNA.
So it's like a key fitting into a lock.
Yeah, that's a good analogy.
And once the receptor is bound to DNA, then it can start to control which genes are turned on and off.
Exactly.
So you mentioned earlier that thyroid hormone receptors can either activate or repress genes. How does that work?
So it all depends on what other proteins are bound to the receptor.
Okay.
So in the absence of thyroid hormone,
The receptor is usually bound to these proteins called core pressors.
and these core pressors, they basically block the receptor from activating genes.
So it's like they're putting a blanket over the genes?
Yeah, kind of. They're preventing the genes from being expressed.
Okay.
But when thyroid hormone binds to the receptor, it causes a change in the shape of the receptor. And that change in shape, it causes the core pressors to fall off.
Oh, so it's like the blanket is lifted.
Exactly.
And then now the genes can be expressed.
Right. And in some cases, the binding of thyroid hormone to the receptor can actually cause the receptor to recruit other proteins called coactivators. And these coactivators, they actually help the receptor to activate genes.
So it's like they're turning up the volume.
Yeah, exactly.
Wow, this is incredible. So, we've been talking a lot about these different receptors and how they control gene expression. But let's shift gears for a moment and talk about another type of receptor called glycine receptors.
Sure.
So glycine receptors are really important for nervous system function. They're involved in regulating things like muscle movement and pain perception.
Yeah, they're essential for maintaining a balance in neuronal activity.
Okay.
Glycine receptors are actually a type of ion channel.
Okay.
So they're these little pores in the cell membrane
Right.
that allow ions to pass through.
Ions are just charged atoms.
Right. So in the case of glycine receptors, the ion that passes through is chloride.
Chloride.
Yeah. And when chloride ions flow into the cell, it causes the cell to become hyperpolarized.
Okay.
And what that means is it makes it harder for the cell to fire an electrical signal.
So it's like it's calming the cell down.
Exactly.
Like putting on the brakes.
Right.
And this is really important for preventing things like seizures.
Oh, wow.
So glycine receptors are really essential for maintaining normal nervous system function.
So we've got, thyroid hormone receptors that control gene expression, and we've got glycine receptors that regulate neuronal activity. It's amazing how these different receptors can have such a profound impact on our bodies.
Yeah, it really is. And you know what's even more amazing is that we're just scratching the surface of what we know about these receptors. There's still so much that we don't understand.
Right. It's like we're exploring a whole new world.
Okay.
And as we learn more about these receptors, we're going to be able to develop new and better treatments for a wide range of diseases.
Absolutely.
Well, this has been a fascinating discussion.
Yeah, I've really enjoyed it.
Me too. And you know what's so cool about all of this is that we're seeing these common themes across these different systems,
Right.
like the importance of molecular interactions and conformational change and the delicate balance between activation and inhibition. Yeah, it's really remarkable. It's like there's this underlying language of biology that's shared by all living things.
That's a great way to put it. So I guess the takeaway message here is that biology is really, really complex.
Yeah.
But it's also really, really beautiful.
Absolutely. And it all comes down to these tiny little interactions between molecules.
It's like a whole hidden world that we're just starting to glimpse.
Exactly. And as we learn more about this world, we're going to be able to develop new and better treatments for all sorts of diseases.
Yeah, we talked earlier about how cancer cells can actually hijack these checkpoints and basically tell the immune system to leave them alone. But now scientists are turning the tables right. They're using checkpoint inhibitors to unleash the immune system and allow it to attack those cancer cells.
Yeah, that's right. Checkpoint inhibitors have really revolutionized cancer treatment. They've been incredibly effective for some patients, but there are still some challenges.
Right. I mean, you're basically taking the brakes off the immune system. So there's always the risk of side effects.
Yeah, absolutely. One of the biggest challenges with checkpoint inhibitors is that they can sometimes cause autoimmune reactions.
Right. So the immune system starts attacking healthy tissues.
Exactly. And that can be really serious. So researchers are working really hard to try to find ways to minimize those side effects. One approach is to develop more targeted therapies that only block the checkpoints on the T cells that are actually attacking the tumor.
Right. So you're not just unleashing the entire immune system.
Exactly. You're trying to be more precise. And another approach is to combine checkpoint inhibitors with other therapies, like chemotherapy or radiation, to try to get a more synergistic effect.
That's so cool. And you know what's really exciting about this is that we're just at the beginning of this journey.
Right.
There's so much more to learn.
Yeah, absolutely. We're learning more and more about the immune system every day, and I think that's going to lead to even more breakthroughs in the years to come.
This deep dive has been amazing. We've gone from, T cell checkpoints to thyroid hormone receptors to glycine receptors, and it's just been incredible to see how all of these different systems work together.
Yeah. I agree. It's really been a fascinating journey.
And you know what's so cool about it is that we're seeing these common themes, like the importance of molecular interactions.
Yeah.
And conformational change and the delicate balance between activation and inhibition.
Yeah, that's right. It's like there's this underlying language of biology that's shared by all living things.
It's incredible. Well, this has been an absolute pleasure.
Yeah, likewise.
And to our listeners out there, keep exploring, keep asking questions, and never stop learning, because the world of biology is full of amazing discoveries just waiting to be made.
Well said.
Until next time.